Welcome to the deep dive. Today we're taking a fascinating plunge into the world of genetics, specifically looking at those intriguing patterns of inheritance that don't quite play by Mendel's classic rules. You know, the ones that add surprising twists to how traits are passed down, almost like nature's little exceptions to the rule book.
That's absolutely right.
Yeah.
While Mendelian inheritance gives us a crucial foundation, the idea that your genes directly dictate your traits, that they're passed on unaltered, they segregate neatly, sort of independently, like you said, like shuffled cards. Well, many genes in eukaryotic species actually deviate from those fundamental principles.
Okay.
So today, we'll explore some of the most compelling ways these rules are beautifully, surprisingly broken.
And our source material for this deep dive is a chapter from Robert J. Brooker's Genetics, Analysis, and Principles, the 7th edition. We're going to unpack some truly mind-bending concepts. From traits influenced purely by the mother's genetic contribution to gene expression changes that don't even alter the DNA sequence itself. And even genes found outside the cell's main command center. Get ready for some aha moments, because this is where genetics gets really interesting.
It really does. It shows us just how intricate life's blueprints truly are.
Okay, first up, let's talk about something called maternal effect.
Right.
Now, you might assume an offspring's traits are all about its own genes. You know, a mix from both parents.
That's the standard thinking, yes.
But here, the mother's genotype completely determines the offspring's phenotype. It really challenges that first Mendelian idea that the offspring's own genes directly influence their traits.
It's quite profound, actually. It demonstrates that some of life's most fundamental instructions are, well, preloaded by the mother. The genes of the father and even the offspring themselves are surprisingly irrelevant to the phenotype you actually see in these cases.
Wow. So how does that work?
Well, the phenomenon is explained by what happens during oogenesis, or egg development. The mother's specialized nurse cells that surround the developing egg produce vital gene products, things like mRNA or proteins. These are then transferred and accumulate in the developing oocyte, the egg cell. And these products persist after fertilization and crucially influence the very early stages of embryonic development. They literally set the stage.
So they're kind of like instructions left behind in the egg cytoplasm?
Exactly. Preloaded instructions.
And the classic example of this is the water snail, Limnaea peregrina.
Ah, yes, the coiling shells.
Right. Their shells coil either to the right, which is called dextral and is dominant, or to the left, which is sinistral. What's wild is how their generations played out in early experiments.
Yeah, those experiments were key.
So when a true breeding dextral female was crossed with a sinistral male, all the first-generation offspring were dextral. That seems normal enough.
Seems Mendelian so far, right?
But get this. In the reciprocal cross, a sinistral female with a dextral male, all the first-generation offspring were sinistral.
Even though they had the dominant D allele from the father.
Exactly. It's completely counterintuitive based on Mendel.
That result clearly contradicted Mendelian predictions, where you'd expect the offspring to reflect their own inherited genes, specifically that dominant D allele. What was truly remarkable was the F2 generation. Even though their individual genotypes showed the expected Mendelian 1.2.1 ratio.
Sure, DD, DD, and DD.
Right. Despite having the genetic potential for both coiling types, they all showed the dextral trait, reflecting the F1 mother's genotype.
Because the F1 mothers were all DD from the first cross.
Correct. And the D mother provides the dominant D gene product to the egg, regardless of which allele she passes on to that specific offspring. It was Alfred Sturtevant who later proposed this maternal effect explanation. The mother's genes specifically produce products that dictate the orientation of the mitotic spindle during the 2 to 4 cell stage.
Just the very first couple of cell divisions.
Yes, that initial cellular orientation determined by the mother's gene products dictates the entire body plan and, in this case, the shell coiling direction.
So the coiling direction isn't about the snail's own DOD allele at all.
It's not for its phenotype, no.
Yeah. But about what gene products its mother's nurse cells supplied to the egg. It's like the mother is pre-programming the fundamental blueprint.
That's a great way to put it. And we see similar maternal effect genes in other organisms too, like Drosophila melanogaster, the fruit fly.
Oh, yeah.
They're crucial there for establishing major body axes, like the head-to-tail orientation. The bicoid gene product, for example, is supplied by the mother and determines where the anterior, the head end of the embryo will form.
So if there's a problem with those maternal genes.
Right. Defective maternal effect alleles often have really dramatic and often lethal consequences because they disrupt these fundamental early developmental processes. The embryo might not even form correctly from the start.
Wow. Okay. That's a big departure from Mendel. All right, moving on to another non-Mendelian pattern that really highlights the complexity of genetics.
Epigenetics.
Yes, epigenetics, a very hot field. This is where genes or chromosomes get modified in a way that changes their expression, but the underlying DNA sequence itself remains completely unaltered. It's like adding sticky notes or bookmarks to the DNA instruction manual.
That's a good analogy. And this is a crucial distinction. These epigenetic modifications, these marks, are often fixed for an individual's lifetime, influencing their phenotype.
Okay.
But they aren't necessarily permanent over many generations. They challenge that second Mendelian idea that genes are passed unaltered.
Because the expression is altered, even if the sequence isn't.
Exactly. The gene's activity is being changed in a way that can be passed down to daughter cells during mitosis. A key example is dosage compensation.
Dosage compensation, right?
Yeah.
Think about it. Human females have two X chromosomes, XX, and males have one X and one Y, XY. So females have potentially double the dose of genes on the X chromosome compared to males. How does the cell balance that out?
That's the problem dosage compensation solves, and different species have evolved incredibly diverse and quite clever ways to achieve this balance.
Like what?
Well, for example, in placental mammals, like us humans, one of the female's two X chromosomes is largely inactivated early in development.
Turned off, basically.
Pretty much. This is called X chromosome inactivation, or XCI. And in humans, this inactivation is random. In some cells, the maternal X is off. In others, the paternal X is off.
But not always random.
No. In marsupial mammals, like kangaroos, it's not random at all. The paternally derived X chromosome is always the one that gets inactivated.
Huh. And what about other animals?
It gets even more varied. In Drosophila, the fruit flies we mentioned, the male actually doubles the expression of most of his single set of X-linked genes.
So they ramp up the male X instead of shutting down a female X.
Correct. And then in C. elegans, a type of nematode worm, the X hermaphrodite decreases the expression of genes on both X chromosomes by about 50%.
Wow, so it's a dampening effect across both X's there.
Yeah.
Fascinatingly different strategies.
It really is. But the most studied example, particularly in mammals, is definitely X chromosome inactivation. Back in 1961, Mary Lyon proposed that one X chromosome in female somatic cells becomes highly condensed into a structure known as a Barr body.
Ah, the Barr body. I remember that from biology class. It was identified earlier, right?
Yes, by Murray Barr and Ewart Bertram in 1949. They noticed this dense blob in the nucleus of female cat cells, but not male cells. Lyon proposed it was the inactivated X.
And that condensed structure is key.
Absolutely. Its compact nature means most of the genes on that chromosome cannot be accessed by the cell's machinery to be transcribed and expressed. They're effectively silenced.
And this explains the famous calico cat.
The perfect visual example.
It's like your cat is a living, purring genetics textbook right there on the sofa.
It kind of is.
Only female cats can typically be calico because the gene for orange or black fur pigment is on the X chromosome.
Right. And since XCI is random,
different patches of cells in the developing embryo randomly shut down, either the X carrying the orange allele or the X carrying the black allele.
Exactly. Leading to that beautiful mosaic pattern of orange and black patches. Males usually only have one X, so they're either all orange or all black, unless they have a rare XXY condition.
And was this XCI hypothesis tested experimentally?
Oh, yes, very elegantly. A key study in 1963 looked at an X-linked enzyme called G6PD, which exists in different forms. What they found was that while a tissue sample from a heterozygous female would show both forms of the enzyme, if they took individual cells from her and grew them into clones, so groups of cells all descended from a single original cell, each clone expressed only one form of the enzyme.
Proving that the decision to inactivate one X or the other was made early on in that original cell.
Precisely. And that decision was stably inherited by all its daughter cells through mitosis. It proved XCI happens early and is maintained.
So it sounds like there must be a master control switch for this X inactivation. How does the cell manage to coordinate such a complex shutdown of a whole chromosome?
You're right, there has to be a control center. The entire process is coordinated by a specific region on the X chromosome itself, called the X inactivation center, or ZEEC.
Easy. Okay.
And XCI occurs in 3 critical phases. First, there's nucleation. This happens during embryonic development, where somehow one X is chosen to remain active and the other is marked for inactivation. The mechanism of choice is still quite complex.
Then what?
Second, spreading. Inactivation begins at the ZEEC and then spreads out along the chromosome in both directions, like a wave of silencing.
Like zipping it up?
Kind of, yeah. And finally, maintenance. The now inactivated X chromosome, the bar body, is maintained in this silent state throughout the individual's life and faithfully replicated in this condensed form whenever the cell divides.
So it's a permanent decision for that cell lineage.
Largely, yes. Although what's particularly interesting is that not all genes on the inactivated X are silenced. Some manage to escape inactivation.
Really?
Yes, especially those in regions called pseudo-autosomal regions, which are areas that are also present on the Y chromosome and are important for XY pairing during meiosis. So it's mostly silenced, but not 100%.
Okay. Epigenetics is definitely adding layers of complexity.
Absolutely.
Our next epigenetic twist is called genomic imprinting. This sounds intriguing.
It really is. This is where a gene is essentially marked or stamped based on whether it came through the egg or the sperm. So whether it was inherited from the mother or the father.
A parental mark.
Exactly. And that mark is remembered throughout the organism's life, determining whether the copy of the gene inherited from the mother or the copy inherited from the father gets expressed.
So only one copy is active, depending on the parent.
Precisely. This leads to what we call monoallelic expression, meaning only one of the 2 alleles, either the maternal or the paternal one, is actually expressed in the offspring's cells.
Okay, so again, standard Punnett squares won't cut it here.
Definitely not. You need to know not just which alleles the offspring inherited, but crucially, which parent each allele came from, because that determines if it's switched on or off.
Could you give us an example?
Yeah, a great example is the IGF-F2 gene in mice. It codes for an insulin-like growth factor, which is really important for growth.
Okay, IGF-F2.
Now, in mice, only the paternal copy of the IGF-F2 gene is normally expressed. The maternal copy is silenced or imprinted.
So if a mouse inherits a functional, working EGF2 allele from its father and a non-functional, mutated EGF2 allele from its mother,
it will be normal size because the functional paternal allele is expressed and the non-functional maternal one is silenced anyway.
right, but what about the other way around?
Conversely, if the mouse inherits the functional IGF-F2 allele from its mother and the non-functional IGF-2 allele from its father, it will be dwarf. Because in this case, the paternally inherited IGF-2 allele is the one that's supposed to be expressed, but it's defective. And the maternal functional IGF-2 allele is silenced by imprinting.
Wow. So even with the exact same genotype heterozygous, one good copy, one bad this phenotype, normal or dwarf, depends entirely on which parent gave which copy.
That's the essence of imprinting. The parent of origin dictates the outcome.
It's fascinating how this happens. What's the mechanism behind this marking process? How does the cell know?
The marking process itself primarily involves DNA methylation.
Adding those little chemical tags to the DNA.
Exactly. Methyl groups are added to cytosine bases in the DNA, usually at specific regulatory regions near the imprinted gene. One such key region is called an imprinting control region, or ICR. This marking, this methylation pattern, is established during gametogenesis, so during the formation of sperm or eggs.
Okay, so it's set in the parent's reproductive cells.
Yes. And then, once that imprint is established in the sperm or egg, it's maintained in the somatic cells of the offspring throughout its development and life.
But what happens in the next generation? Does the mark just pass on directly? If a female inherits a paternally marked gene, does she pass it on as paternally marked?
Ah, no. That's where it gets even more intriguing and critical for the process to work over generations. In the germline cells of the developing offspring, the cells that will eventually produce its own gametes, the old imprint is erased.
Wiped clean.
Wiped clean. And then a new imprint is established based on the sex of that individual.
Okay, walk me through that with the IGF-F2 example.
Right. So if the mouse is female, regardless of whether she inherited an active paternal IGF-2 or an inactive maternal IGF-2, in her developing eggs, all IGF-2 alleles will be marked with the maternal imprint, meaning they will be transcriptionally inactive.
So she resets it to maternal off.
Correct.
Okay.
And if the mouse is male, regardless of his inheritance in his developing sperm, all IGF-2 alleles will be given the paternal imprint, meaning they will be transcriptionally active.
So he resets it to paternal on. It's like a sex-specific reprogramming cycle.
Exactly. This erasure and reestablishment ensures the correct imprinting pattern is passed down according to the parent's sex. It acts as a crucial reset mechanism each generation.
The system seems quite complex. Does it ever go wrong in humans?
Unfortunately, yes. Genomic imprinting plays a significant role in certain human genetic diseases.
Such as?
Well, the classic examples are Prader-Willi syndrome, PWS, and Angelman syndrome, AS. Both of these distinct disorders are often caused by the exact same small deletion on human chromosome 15.
Same deletion causes two different syndromes. How?
It depends on which parent you inherit the deletion from. If the chromosome 15 with the deletion is inherited from the father, it results in Prader-Willi syndrome. This is because the deletion removes several genes that are normally expressed only from the paternal chromosome in that region, like SNRPN and NDN. The maternal copies are imprinted, silenced.
Okay. And Angelman syndrome?
Angelman syndrome occurs if the same deletion is inherited from the mother. In this case, the deletion affects a different key gene in that region, UBE3A, which is normally expressed primarily from the maternal chromosome in the brain. The paternal copy is imprinted, silenced, in the relevant cells.
So that's a really striking example of how the parent of origin, not just the presence or absence of the gene sequence itself, dictates the disease outcome. Same deletion, totally different consequences based on inheritance.
Absolutely. A clear, albeit tragic, demonstration of imprinting in humans.
All right, so we've had maternal effect, epigenetics, with XCI and imprinting. Well, what's next?
Finally, we venture outside the cell nucleus entirely.
Outside the nucleus, where else is there genetic material?
We're talking about extranuclear inheritance, sometimes called cytoplasmic inheritance. This is where genetic material influencing traits isn't located on the nuclear chromosomes at all.
Okay, this really breaks Mendel's rules, especially segregation, right? Because these genes aren't on chromosomes that pair up and split during meiosis.
Yeah, exactly. The segregation patterns are completely different. The most significant examples involve genes located within two types of organelles in the cytoplasm, mitochondria, and, in plants and algae, chloroplasts.
Mitochondria, the powerhouses of the cell. And chloroplasts for photosynthesis. They have their own DNA.
They absolutely do. These organelles have their own genetic material, typically small circular chromosomes found in regions inside them called nucleoids. They look a lot like smaller bacterial chromosomes, actually.
Interesting. How much DNA are we talking about?
Well, in humans, mitochondrial DNA, or mtDNA, is quite small, only about 17,000 base pairs long. It contains a relatively small number of genes, 13 that code for proteins essential for oxidative phosphorylation, the main energy-producing pathway.
Okay.
Plus the genes for ribosomal RNAs and transfer RNAs needed to actually make those proteins inside the mitochondria.
Only 13 protein-coding genes. Mitochondria seem more complex than that.
They are. It's important to remember that most of the proteins needed for mitochondrial structure and function, hundreds of them, are actually encoded by genes in the cell nucleus. Those proteins are then made in the cytoplasm and imported into the mitochondria.
Ah, okay. So it's a collaboration between nuclear and mitochondrial genes. What about chloroplast DNA?
Chloroplast DNA, cpDNA, is generally larger than mtDNA, often carrying more genes, maybe 100 to 200. These include many genes required for photosynthesis and other chloroplast functions. But again, many chloroplast proteins are also encoded by nuclear genes.
So how are these organelle genes passed down from one generation to the next if they're not segregating with nuclear chromosomes?
In most animals and plants, it's through maternal inheritance.
Primarily from the mother. Why?
It comes down to the gametes. The female gamete, the egg cell, is typically very large and contributes the vast majority of the cytoplasm to the resulting zygote after fertilization. And that cytoplasm contains all the organelles, including the mitochondria and, in plants, chloroplasts.
Right, the egg is huge compared to the sperm.
Exactly. The male gamete, the sperm, is usually very small and contributes little more than its nucleus. Sometimes a few mitochondria might get in, but they're often actively destroyed or vastly outnumbered. So effectively, almost all of your mitochondrial DNA, for instance, comes directly and exclusively from your mother, her mother, and so on, up the maternal line.
Is there a classic example for this, like the snail for maternal effect?
Yes. Carl Correns' work back in the early 1900s with the 4 o'clock plant, Mirabilis jalapa.
What did he find?
He studied plants with different leaf pigmentation. Some had all green leaves, some had all white leaves, and some were variegated, meaning they had patches of green and white. He found that the phenotype of the offspring's leaves depended solely on the phenotype of the branch from which the female gamete, the ovule, came. The pollen source, the male parent, had no influence on leaf color.
So if the flower providing the egg was on a variegated branch.
Then her offspring could be all green, all white, or variegated themselves. This was because the chloroplasts, which determine leaf color, green have normal chloroplasts, white have mutant ones, were inherited exclusively through the cytoplasm of her egg cell.
And that variation, getting green, white, or mixed offspring from a mixed mother, ties into another concept, right? Heteroplasmy?
Precisely. Heteroplasmy is the state where a cell contains a mixed population of organelles, in this case chloroplasts. Some have the normal gene, leading to green. Some have a mutant gene, leading to white.
So the variegated plant is heteroplasmic.
Yes. Its cells started out with a mix of normal and mutant chloroplasts. As the plant grows and its cells divide, these chloroplasts are sort of randomly distributed to the daughter cells. Sometimes, by chance, a daughter cell might end up with mostly or only normal chloroplasts, leading to a patch of green tissue. Or it might end up with mostly or only mutant chloroplasts, leading to a white patch. It's this uneven segregation during cell division that creates the variegated pattern.
Like a random sorting process within the cell lines?
Exactly, a random segregation of organelles during mitosis.
And this obviously has huge implications for human health when we talk about mitochondrial DNA.
Absolutely. Many human diseases are now known to be caused by mutations in mitochondrial DNA.
And since mtDNA is maternally inherited,
These diseases follow a strict maternal inheritance pattern. An affected male cannot pass the condition to his children, but an affected female will pass the mitochondria and potentially the mutation to all of her children, though the severity might vary.
what kind of diseases are we talking about?
They tend to be chronic, often degenerative disorders that primarily affect tissues and organs with very high energy demands because mitochondria are all about energy production. So nerve cells and muscle cells are often heavily impacted.
Can you give some examples?
Sure. Leber hereditary optic neuropathy, or LHON, which causes progressive vision loss, is one well-known example. Others can cause various forms of muscle weakness, deafness, heart problems, or neurological issues.
And does heteroplasmy play a role in the severity of these human diseases too?
Yes, a very significant role. Just like in the variegated plants, human cells can be heteroclasmic for mtDNA mutations. The clinical symptoms and their severity often depend on the ratio of mutant to normal mitochondria within the cells of critical tissues. If the proportion of mutant mtDNA is below a certain threshold level, the person might be asymptomatic or only mildly affected.
But if it's above that threshold,
Then they might exhibit severe symptoms. This explains why, even within the same family inheriting the same mtDNA mutation, there can be such wide variation in disease presentation. It depends on that random segregation process during development and the resulting percentage of mutant mitochondria in key tissues.
it adds another layer of complexity to predicting disease.
It certainly does.
And thinking bigger picture, the whole idea of organelles like mitochondria and chloroplasts having their own DNA, that connects to a really fundamental evolutionary concept, doesn't it? The endosymbiosis theory.
Yes, it's one of the cornerstones of modern biology. This widely accepted theory proposes that mitochondria and chloroplasts were once free-living bacteria that were engulfed by ancient, larger prokaryotic or early eukaryotic cells.
Engulfed but not digested.
Right. Instead of being destroyed, they established a symbiotic relationship with the host cell. Chloroplasts are thought to have evolved from cyanobacteria, which could photosynthesize.
And mitochondria.
Mitochondria are believed to have originated from a type of bacteria known as purple bacteria, which were efficient at cellular respiration.
And the evidence for this?
The evidence is quite strong. Their DNA, their ribosomes, their structure, they all resemble bacteria much more closely than they resemble the eukaryotic cell components around them. Their genes are organized more like bacterial genes.
But it was a beneficial partnership.
Hugely beneficial.
Yeah.
The host cell gained the ability to photosynthesize in the case of chloroplasts, or to produce ATP much more efficiently using oxygen in the case of mitochondria. The endosymbiont gained protection and resources.
And over evolutionary time.
Over vast stretches of time, many of the original bacterial genes from the endosymbiont were either lost or transferred to the host cell's nucleus. That's why nuclear genes now code for so many organelle proteins. But they retain their own distinct genomes as a legacy of that ancient symbiotic event. It's a remarkable story encoded within our very cells.
So we've just journeyed through this really incredible world of non-Mendelian inheritance. From the surprising influence of a mother's genotype pre-programming her offspring's physical traits in maternal effect,
Right.
to the dynamic, non-sequence changes of epigenetics, like XCI creating patterns in calico cats, or imprinting deciding which parental gene copy gets hurt.
Yeah, the dimmer switches and
Yeah.
parental stamps.
And finally, exploring the ancient stories held within our cells' very own powerhouses, the mitochondria and chloroplasts, passed down primarily through the maternal line.
These patterns truly remind us that genetics is far more nuanced and interconnected than the initial simpler Mendelian models might suggest. It's really a continuous story of discovery, with each new understanding revealing deeper layers of complexity and, honestly, wonder in the mechanisms of life.
Yeah.
Understanding these deviations from Mendel's basic rules is absolutely key to grasping the full picture of how traits are inherited and expressed in the real biological world.
And it truly shows how adaptable and, I guess, creative life is in passing on its blueprints.
Yeah.
What stands out most to you from this deep dive? For me, maybe it's just how something as seemingly simple as which way a snail's shell coils can reveal such a fundamental non-Mendelian rule of inheritance. Or maybe how your cat's fur pattern is this living testament to epigenetic mechanisms happen constantly.
Those are great examples. For me, it's perhaps the elegance of the solutions, like dosage compensation. So many different ways to solve the same fundamental problem across species. These examples all highlight the incredible range of ways genetic information is managed and expressed, shaping every living thing around us. They really push us to think beyond simple dominant and recessive alleles.
Yeah, definitely makes you wonder, doesn't it? What other hidden layers of genetic control are still out there waiting for us to discover?
Always more to learn.
Well, thank you for joining us on this deep dive into non-Mendelian inheritance. We hope you, our listener, feel a little more well-informed and perhaps a little more amazed by the intricate dance of genes. And to our dedicated listener, we extend a warm thank you for being part of the Last Minute Lecture family. We appreciate you.